Today I will be talking about the Immune Epitope Database and Analysis Resource for our AMP lab activity. So on this website, for this specific lab, we will be just using our left side of the website and the middle section of the website. Brief summary of what this website is about, and below we see the upcoming events. You can also register to any virtual user workshop, and they also have more information of specifically how epitope analysis for SARS-CoV-2 database can be used for this specific organism. And then we have our summary metrics, which gives you a summary of the database. And the importance of this website is it was created to assist biomedical researchers in the process of developing new vaccines, diagnostics, and therapeutics. And it provides analysis of epitope analysis and prediction tools. So here we have the epitope box. So we know the epitope means it's parts of the antigen molecule to which an antibody attaches itself to. And for today's lab, we will be putting any. And the epitope source, so we're going to be putting the organism, right? It can be influenza, peanut, but today we will be putting SARS-CoV-2. And you could be putting the antigen, but in this lab, we will be hovering over that and not doing that one. And then we have host. So we are going to be choosing Homo sapien, but there is a few other choices you can make, and if you want to navigate the website by yourself, you can also go to find, and you can put a dog or something else. And we have the assay. So we have the assay. We have T cells, B cells, and MHC ligand. And if you just want, information from T cells and B cells, so these white blood cells, and just choose T cells and B cells or whichever one you want. But for today, we will be choosing all of them for this lab. And also, it gives you the choice of your outcome choice. And for today, we will be using positive and negative. What does positive and negative outcome mean? So the positive data is when the assay responds to the epitope and generates a response against it. And if it's negative, this means that the base cell does not generate against that epitope. That's pretty. And then we have the MHC restriction. So we have class 1 and class 2, or non-classical, right? MHC restriction actually stands for major histocompatibility complex. In the human, it's the human leukocyte antigens, which is located on the surface of every cell in the human. And MHC just helps our white blood cells to distinguish our healthy and our infected cells. And I mentioned there is class 1 and class 2, but for today we will be just choosing any. And disease, in this case it is infectious, but we can put any as well. There's just a few more options, and of course there is more search, right? A search box. And then we can go ahead and click search. And it can sometimes happen that it does not work right away. So you can just go back and click up here to home, and then try to do it once again. I found that sometimes just works best. And you may have to just be a little patient with this website. And there we go. And here on the left side, we will be seeing the pending filters. And this is what we saw on the home page, and it's all here again. Filter options, epitope source, receptor, assay, and just make sure that the outcome is positive and negative again. And MHC restrictions, host, disease. And now here, on what we see that it's new, we have these tabs with talks about the epitopes. And this is the epitope, the classification of each one, the antigen, organism, and there's reference and assays. And then we have antigen. Antigens, there is a list of them. Assays, we have here more. In the assays, it really does break down more information about each of these epitopes. So it gives you a reference, epitope, host, immunization, assay antigen, antigen epitope relation, and also there is these type of receptors and then references. If you have just a specific interest in all these references, you can always just click this link and it will take you to that reference. So now going back to antigens, actually, we are going to be hovering over the immuno browser. So here, if you hover over this little graph, it just gives you an overview of that immuno browser. And what we're going to do is now click that little graph and it will prompt a new tab. And you will have to be patient with the new tab. It's going to take a while. It may take a while. There we go. Now it's prompt. And then let's see if we get this one back. Yeah, it's back. Okay. So let's go back. Okay. So here a little overview about the immuno browser. So here we will have the name of our protein, which is going to be SARS-CoV-2 spike glycoprotein. Here we will see the information. So these are two graphs. We have, and going down here, we will be looking, these are the epitope sequence, and this is the epitope ID, the map positions, right? And subjects tested, subjects responded, assays positive, negative, and responded frequency. And I will talk about how this information became this. So starting with this first graph, the response frequency graph. So this graph informs us about how immunogenic a specific residue is. In other words, a higher residue means our antigens have been triggered to form a response. In the x-axis, we have it just shows the residue numbers of the protein, and the y-axis indicates the response frequency. And what we see here in this graph is the confidence interval of the response frequency. And it's found this response frequency, so RF, so it's responded subjects divided by the total tested subjects. So going back down here, we see subjects tested, subjects responded, and then we have, respond frequency, right? This information just allows us to correct for the sample size. And it shows a range of possible values that will contain the real response frequency value for 95% of the time. And going down here, we have the epitope assay counts, right? So what this graph shows us is how many times a specific amino acid is present in a positive or negative assay. And it gives us an idea of the immunogenicity from the region. And for this specific lab, we have a residue position from 300 and 600. And we have to select a variant, and for today's purposes, I'm just gonna choose B4, BA 4.6. There we go. And then it, as I mentioned, takes a little bit to catch up, but let's go ahead and we can update the display. And then here we see it zoomed into this residue positioned section, right? So we have 300 to 600. And we are going to start seeing that upper bound and lower bound. You can also, a few features that you will see is that you can download, save, and annotate on this graph if you are willing to, and print from here as well. And You can also choose just to see the upper bound. You just need to click on the on yeah, here, and then you will only be able to see the upper bound. That helps you. I'm gonna bring it back, and also here we have the negative and the positive, right? And positive will be blue in this the epitope essay count, and then the negative will be the red. And we are going to be looking for the upper bound, so one of the highest peaks. And here we have this high peak, and it will show you here the actual mutation. And we have here F486V, and then here is where you want to take those screenshots. And so you can save and find those upper bounds. And sometimes there are other, upper bounds, but there is no mutation. So this may happen to you. Here we have 487. If we see here, at 487, we have a spike, and we can see here 487, right? So lightly. Sometimes it may not prompt for you to see that mutation, so sometimes you just kind of have to zoom in and then figure it out from here. I mean, there we go. You see, it provided for you, which is F486V. So those are two already mutations, and then you just want to screenshot it to have that information. Okay, and also you will have to choose a different variant. So I'm gonna choose a different variant just to give you guys a idea. So we have, different variant. Positions, the start and the end of the position is gonna still be the same. And it already updated, but just to make sure, just update the display. Sometimes it may just take a while. And for this specific variant, we only see 2 mutations. But anyways, that is all.